Welcome to one of the past HSC exam question videos. In this video we're going to cover this exam question, which comes from the 2011 paper, and it's question 24 worth 5 marks all up. The actual question is 3 parts to it, and I'll read them out in a second. The first part says, name a viral disease that is controlled by the use of vaccines. The second part is, the flow shows the steps involved in the preparation of a vaccine to prevent a viral disease. Isolate virus, break up virus particles, retain protein code, discard nucleic acid, suspended protein in saline solution, viral vaccine. Outline how a vaccine that is prepared in this step will produce immunity to a specific viral disease with 2 marks, and explain one procedure in the flowchart that ensures the safety of the prepared vaccine, also worth 2 marks. So when you're ready, pause the video and attempt the question, and afterwards I'll go through a marking guideline and some sample answers in terms of how you could have answered that question correctly. Alright, so when you're ready, pause the video. Welcome back. Alright, so in this case, the first one, name a viral disease that is controlled by the use of vaccines. And so it's worth 1 mark, so all you have to do is name, which means literally just name it. The actual dot point or the actual marking guideline says you need to identify that one, which is fair enough. Here I've got 4 different ones, and one of them is not actually a virus. It says you need to have a viral disease, so a virus. And this one, the one that is not, this one is a bacterial one. This one is bacterial. So if you said diphtheria, you would have not gotten a point. You need to say polio, measles, or smallpox. And the one that is 100% controlled, that one's been almost eradicated more or less. Yeah, it has been eradicated, smallpox. And the other ones still exist, but they're more or less controlled. Diphtheria is bacterial. Even though it's controlled, it's not viral. Alright, so any of these would have done. So I just wrote smallpox is one disease caused by a virus that has been controlled using vaccines. And that will get you your mark. Now for the second one, so outline how vaccines prepared in this way produce a specific viral immunity to a specific virus. So what I should have kind of written is a couple steps. So first of all, this protein, so we have to retain the protein code here, and this protein code contains these antigens. So the virus has these antigens on it, on the protein code. So by having this in our vaccine, this will activate the immune response if we inject it into a person, right? Because they have these antigens on the protein code. If they activate an immune response, that means that we're going to have afterwards, after the primary immune response is over, we're going to have these memory cells being produced. And these memory cells means that whenever the pathogen re-enters our blood, we've got the secondary immune response that really quickly kills off the pathogens in the future, which means now we have the immunity. Right? So this is basically what I kind of should have written to get your full marks, because it says, yeah, indicate one main feature of the viral vaccine. So I chose a retained protein code, and link it to a named immune response. And that was a secondary immune response. So what I wrote is, the protein code contains antigens that will activate the immune response when injected into our body. Right? So these got these antigens on the protein code. So that's going to get me probably 1 mark already. But now I want to make sure I link it all to the immune response itself. T and B memory cells will appear in the blood after the primary immune response is finished. So these memory cells will be there after that first infection. And these memory cells will activate the secondary, more powerful immune response once the same antigens re-enter our body. So once it comes back, then it's going to be quickly killed, which means we have immunity. And the reason why it's going to be quickly killed is because the antibodies that are produced from secondary immune response will quickly fight off any subsequent infection. Right? So if you say the first 2 statements, that should already be enough to get you 2 marks. But the last one was just added to make sure. Right? But the idea would be, you say that the protein code contains antigens, and you quickly describe how these antigens will cause the memory cells to be produced, and the memory cells will then mean that we have the secondary immune response comes in the future, and that means no more infection. Alright, so the last one was this one, and it says explain one procedure in the flowchart that ensures the safety of the prepared vaccine. You could have talked about 2 things. You could have talked about either the discard the RNA DNA part, right? Because by discarding that DNA part, this part here, we have no invasion of body cells. They need to have this DNA to be able to invade body cells, right? They need to replicate. So no replication for these viruses. That means they're harmless. If they can't replicate, they're going to be harmless. Another one you could have chosen would be the saline solution. And the reason why that's going to be safe is because that means they're going to have the same salt concentration as the surrounding cells that they've been inside. So if we've got red blood cells, for example, inside our blood, and that means the saline solution, which the protein code is encased in, will be that same salt levels, which means there's no osmosis happening, there's no water going in and out, which means these cells around it are not affected by the change in salt concentrations, because it's going to be the same isotonic salt concentrations. But this is, you choose either one of them, right? And obviously, I guess the most what we've taken would have been this one, discard RNA DNA. And this is the one I'm going to describe in more detail. So what I wrote is, discarding nucleic acid takes away the virus's genetic information, and it needs to reproduce. This means that the virus cannot successfully invade host cells and take it over to produce more viruses, right? Which means, yeah, it can't reproduce, and thereby it'll become harmless. So then, the virus contained in the vaccine is therefore harmless and does not cause disease. So that statement in the end would also be useful. You say it can't reproduce, that's good. And you say because it can't reproduce, it can't cause disease, therefore it's harmless. That's also a good statement to say. And that will get you 2 marks out of 2. Alright, where did that question came from? It came from these dot points. So the first one, A and B, and A and B, they came from this one. It talks about the different types of vaccines that were produced that have to include smallpox, diphtheria, and polio. And remember, diphtheria is the only one here that is a bacterial. The other 2 were virus that were controlled. And outline the ways in which vaccinations prevent infections. That's this part, so we have to outline. And this is also, so this second part as well is connected to that dot point. These both 1 and 2 connected to this one, and the first one, A, was connected to this one. I hope that was useful.